Anyway, good afternoon, everybody. This afternoon, the panel is going to focus on psych, and much like what was done before, we'll just go around the table here for a brief introduction and before we get into the topic. So my name is Murali Gopalakrishnan. I lead our search and evaluation function at AbbVie. AbbVie, as you know, is a pharmaceutical company. We are headquartered in Chicago. Neuroscience is a considerable focus for the company, and we are active in a number of areas, including neurodegeneration, Alzheimer's, Parkinson's, ALS, psychiatry being another focus, and also migraine and pain. So we're broadly covering a number of areas in neuroscience. It's my pleasure to be here, and I'm going to introduce my co-chair, Manuel, as well for brief introductory comments.
Thank you, Murali. First of all, thank you, Leo, for your kind invitation to the meeting. This keeps exceeding expectations every year, so it's wonderful to see a lot of familiar faces here today. My name is Manuel López-Figueroa. I'm a neuroscientist by training and 20 years in venture capital, and as well as in the Pritzker Consortium, which is focused on psychiatric disorders, as well as a chief operating officer at Genomics. I'm going to leave Carlos Buesa.
Okay, thank you, Manuel. I'm Carlos Buesa. I'm founder and CEO of Oryzon Genomics, a company which is developing drugs in epigenetics and concretely in CNS. We are having an asset right now in phase 2 in schizophrenia for negative symptoms and cognitive decline, and phase 3 ready in borderline personality disorders. It's basically a new mechanism of action. It's a chromatin remodeler, an epigenetic enzyme. And we have so far dosed almost 500 people. So it's a very, very safe compound. And we have got a number of very interesting interactions with the FDA in the last months that I think will be part of the discussions today.
Eliseo.
Good afternoon, Eliseo Salinas, head of R&D at Delix Pharmaceuticals, where we develop drugs that grow dendritic spines, and we're going to talk a little bit about why is that important. I've been 34 years in the industry, the last 20 as head of R&D, head of research or clinical of mid-size and small biotech. Very happy to be here today talking about this revolution in neuropsychiatry. I have to disclose that all the revolutions I supported failed, so I'm betting on this one. No.
Hello, everyone. I'm Jens Wendland. I'm the Chief Medical Officer at Kynexis Therapeutics. I'd also like to thank the organizers for allowing me to be on this panel. Kynexis is a clinical stage biotech company that develops a CAT2 inhibitor. CAT2 is the rate-limiting enzyme for an endogenous inhibitor of NMDA and Nic alpha-7 receptors. So basically, by inhibiting this enzyme, we are releasing the internal brake on these receptors. And so our current focus is on developing this asset for cognitive impairment associated with schizophrenia. Just a few weeks ago, we read out our first in-human study where we saw absolutely spectacular results with target engagement across biochemical and ePhys domains, as well as early signals of efficacy on cognition. Based on these results, we are accelerating our first in-patient study, which will begin in the next quarter for CIAS, and also actively pursuing additional indications. And I look very much forward to discussing these and other things today.
Thanks. Laszlo.
Laszlo. I'm with Gilgamesh Pharmaceuticals. We are also a clinical stage pharmaceutical company focusing on psychiatric disorders. We have a suite of programs that range from psychedelic to non-psychedelic programs positioned for both in-office as well as at-home use. We've been around as a company for about 5 years now. And our 2 lead programs are about to read out phase 2A studies in MDD patients first quarter of this year.
So,
thank you, Murali, for including me in the panel. Thank you for all coming. I'm Seth Lederman. I'm the CEO of Tonix Pharmaceuticals, and we are a small but fully integrated pharmaceutical company. We have just been awarded a PDUFA date of August 15th for our lead program, which is a new treatment for fibromyalgia. Fibromyalgia is a chronic pain condition, affects mostly women. There are about 10 million patients in the United States. There hasn't been a new drug in fibromyalgia in over 15 years. So we're very excited about the upcoming potential approval and potential launch. We have a small commercial operation selling 2 small migraine drugs, so we're very involved in pre-launch activities at risk of the approval. Thank you very much.
Vikram.
Congratulations on a PDUFA date. That's great. My name is Vikram. I'm co-founder and CEO of Engrail Therapeutics. Scientist by training, went to the industry side doing drug pricing and launch strategies, then set up a small investment bank, and also been on the venture side for many years. Primary interest in neuroscience. One of the companies I invested in was the first to combine a muscarinic agent with a peripheral blocker. Very compelling pharmacology. Started Engrail 5 years ago. We're clinical stage now, so very exciting. We have a pretty deep pipeline. Lead compound is a subtype selective GABA PAM. It's a best-in-class drug that's in an ongoing phase 2 study for the treatment of generalized anxiety disorder. Again, an indication where no drug's been approved for over 15 years. So we're very excited. The study is going extremely well and should read out at the end of the year. Second compound, which is a dopamine D2/D3 antagonist that we're studying for anhedonic depression. And we have a novel mixed pharmacology drug behind that that addresses the dopamine D2/D3 and serotonin 5-HT1A and 2A receptors. It's a non-hallucinogenic neuroplastogen, so it should be in the clinic in 6 months' time. And we have a rare disease drug, although it's preclinical in development, has shown life-changing effects in patients in exceptional use settings.
Thanks, Vikram. So as you see, our panel is very rich, particularly with companies all in the clinical stages. So it's quite interesting to see that. And then there's a diversity of topics that we're going to be covering. So just to put it in perspective here, and this is a schematic that sort of we drew up in a way. As you certainly know that you think about psychiatric diseases, broadly speaking, it's, certainly you can categorize them based on symptomatologies and group them into different buckets as shown here. Psychotic disorders, mood disorders, anxiety and stress disorders. But as you see very clearly, you cannot clearly bucket each of them in a bucket. There is considerable overlap, particularly with many of the domains that we talk about, the transdiagnostic features, for example, and that's got implications as we think about targets, translational approaches, clinical studies, and so on. So we're going to be covering a lot of that during this conversation today. And we're not going to cover every target or every possibility, but we're highlighting a couple of things, particularly that what has happened since the last SACS meeting, which is about a year ago. So what's really happened in 24, we're going to summarize, where the state of developments are, and then have a good discussion on other questions. If there are questions from the audience, we can always entertain them as well. So we'll kick it off today. I'm going to maybe start off with Vikram, who's on the other side, who can maybe share a perspective on what really transformed or revolutionized, if I use that word, in 2024. And then maybe we can open up the discussion and conversation from there. So Vikram, I hand it off to you. What do you think about and summarizing what's going on in the space?
I'm just really excited about what's going on in neuroscience in general, I have to say. The past few years have been very compelling, notwithstanding some of the failures that we've seen recently. But we've seen significant successes as well. I don't know that you can mention 24 without talking about the impact that Corona has had on or will have eventually on schizophrenia patients. I mean, KarXT or Cobenfy, as it's known now, is such a compelling drug. I mean, an average effect size of over 0.6 is very compelling. I don't know that we've seen such effect sizes since the initial approvals back in the day of risperidone and olanzapine. It's the highest effect sizes that we've seen for schizophrenia, should be game-changing, hopefully. Unlike some of the other antipsychotics, the drug was actually relatively well-tolerated. There's no akathisia, no EPS, no frank sedation. There seemed to be, perhaps due to some PK matching, some breakthrough cholinergic-like side effects like nausea and vomiting. But besides that, given the risk-benefit, seems like a phenomenal drug. So I'm excited for patients. I'm actually quite excited for what that drug can do for Alzheimer's patients as well, because it should have a significant improvement in cognitive benefit. And as we know, Alzheimer's patients don't actually die because of the disease. The majority of deaths are because of the side effects, or related issues with Alzheimer's, falls, infections. These are big concerns. So symptomatic improvements that improve cognition can go a long way. So I'm hoping to see this drug actually used even more widely. So I think that was exciting. Caplyta has generated some really exciting data. I mean, it's an old approval, first for schizophrenia and then bipolar 1 and 2. But if you see, they had really good effect sizes in MDD recently, north of 0.5 in 2 studies. And so I'm excited for that drug. Good effect size, safety profile is outstanding for Caplyta, as we all know. Excited for seltorexant from J&J. I think that's a very meaningful drug, and it'll touch on one of the themes that we will talk about in this panel, which is getting much more selective for patient subtypes. So, that was in previously unresponsive patients, but with an insomnia phenotype. And so starting to segment patients really well with that compound. Quite excited for the potential for axons or Auvelity in Alzheimer's patients as well for agitation. Could be the first, if it's approved, non-psychotic mechanism of action for treating agitation. So quite a few. I know I've left out quite a few here, but just these highlights, have been really good for 2024.
Yeah, those are great examples. So thank you for sharing that. One of the other space that comes to mind, and I know Laszlo, you briefly touched on it as well, around what's uncovering new biology and new directions being taken, and the psychedelics, or you call it neuroplastogens, are shedding some interesting light as well. I know these are early days for those programs, but I don't know whether Laszlo would like to elaborate perhaps on emerging biology and emerging aspects that can open up into the next wave of targets, for example.
Yeah, certainly the psychedelics have been, seen a resurgence. And if you look at the clinical data for the companies that have run phase 2 trials, it's all been positive, and the results have been quite robust. So certainly this renewed interest in the space has brought people in, companies like us, to really interrogate these targets comprehensively. There's plenty of room to make improvements on, molecules that have maybe been around for decades, in some cases millennia. So there's plenty of room to make best-in-class molecules based on the pharmacology of these molecules and what we understand about the receptors. But I think it's also important to kind of put these targets in the perspective of what they do at a network, circuit, and whole brain level. So we've really taken sort of a comprehensive approach to this, in the sense that not only do we think about the targets themselves and what the downstream signaling might look like and how you might want to optimize that, but what kind of effects, when you make these modulations and make molecules that interact with the receptor in various ways, what does that have on the network, on the circuit itself? And so we've sought out new tools to interrogate that in a preclinical level, either as cultures in a dish or at a whole brain level, using EEG, neuropixels, techniques like that, to really help us understand the biology at more of a larger scale. And then we also need to incorporate novel, new behavioral assays preclinically as well. These assays that are unbiased, they measure behavior in ways that you really get a comprehensive look at what the effects of the drugs are. When you start to ask questions about making slight modifications to the target and how you interact with it, and then how does that translate into what happens at a network level, what happens at a behavioral level preclinically. And these are the kinds of tools that really sort of help us understand the targets, better help us understand the kinds of molecules we're bringing forward in a much more comprehensive way, so that then we can then say, okay, based on what we understand about, how these molecules behave in vitro and in vivo, how does that now translate into what we see in the clinic? Translation's still a challenge, as everybody knows. Psychiatry, the models are not fantastic. But I think if you can understand how a molecule works at a circuit and a behavioral level, and if you can differentiate various mechanisms at a preclinical stage, then I think you can start to sort of position various molecules for clinical trials that really sort of get at very unique mechanisms. And I think then you have to, it's really important then afterwards that when you go into the clinic, you have a plan in place that really lets you answer key questions. You got to have a good molecule that gets to the target, and you need to know that you're engaging the target, and you have to have biomarkers or subjective effects that really help you characterize what that molecule has done, even early stage in healthy individuals. Like, does the mechanism that you believe in preclinically translate into effects that you expect to see in the clinic, and then ultimately tested in patients?
Yep. Thanks, Laszlo. So if I understood the way you're thinking about it is, and I hear this from some colleagues, right? Think about psychiatry. Well, these are all old targets, right? Muscarinics have been around for how long, right? And you think about other targets. So, I'm not hearing this like a breakthrough to say here is a new area of target biology, but what I'm hearing from Laszlo is like, how are we really applying some of the principles to, study these molecules, understand their biology, and translate that to the clinic. And the refined understanding is what is really propagating and fueling the innovation in the psych space today. I mean, that's one way to do it. Instead, I think when we talked about this before, you are thinking about targets slightly different from a symptomatology perspective or a syndrome perspective, as well as opposed to molecular targets and hitting specific molecular elements. I don't know, do you want to elaborate on that thinking as well so that we can move on to the next topic?
Yeah, thank you. I think many people think of targets in very limited ways, just a molecular target like a muscarinic receptor subtype or something. But and I think an equally rich area for new targets is to think about the rapidly evolving understanding of conditions and syndromes. We work in fibromyalgia is a pain condition, but it's on the suburbs of psychiatry because there's a high overlap with depression and anxiety. But there's a rapidly emerging or emerged consensus that there are now three primary types of pain: nociceptive, neuropathic, and now the newest kind, nociplastic pain, which is now the plenary session of the IASP and other places. And the idea that pain states present as these three different types. So understanding that subdivision, a clinical subdivision of a type of pain, opens up a whole new way of attacking these syndromes. But similarly, the idea that different conditions where you may not be treating the whole condition, for example, Acadia is very successful pimavanserin development for certain symptoms associated with another condition, Parkinson's, like hallucinations and other things. That I think subtyping out these things can be just as important in selecting patients or even even biomarkers in narrowing down a therapeutic development and increasing the possibility of success.
Thank you. Thanks very much. So I think one of the areas that we briefly touched on around innovation is around psychedelics and the sort of what has changed in the space. And there's a whole session after this on psychedelic approaches. So we're not going to get into that topic in any detail, but maybe I'm going to ask Eliseo to kind of comment on how we're thinking about further innovation in this space, the sort of the next generation approaches that can really, push into, various mental health disorders and also other therapeutic uses of such molecules.
Yeah, thank you. I think it's important in the text talk about benefits, therapeutic benefits. And it's important talking about depression, define what are we talking about? Of course, they improve depression, but what are the therapeutic benefits? And for that, if you allow me, I think it's important to look back a few years. The first antidepressant we ever had was electroconvulsive therapy, electroshock, ECT, 1938. The only thing that was used and approved. What did it do? ECT had a single dose effect. You do a shock, patient improved for days, immediate. Number two, but that doesn't cure the depression. You need to give it 3, 4, 5 times, every 3, 4, 5 days. Eventually, you get the patient to remission and you stop. Patient goes home and comes back with a new episode years later. Number two, single effect, but needs to be repeated several times. And number three, a huge disconnect between the stimulus, the duration of the treatment, which is less than 5 seconds, a shock, and then the clinical benefit that lasts for 3 or 4 days with the initial shocks, and then for months or years later, suggesting that there were post-transcriptional effects. 38, 40s, 50s, lots of studies on that. 58 came the first tricyclics, MAO inhibitors in the 80s, SSRIs and SNRIs. Very different mechanisms of actions, all of them, serotonin or adrenaline, this one, that, and we had been in discussions which one is more important and so on and so forth about neurotransmitters. But in terms of benefit, they all do the same thing. No single dose effect. A single dose of any antidepressant does nothing for depression. Number two, you need to give them for days and days and days until you see the first effects 3 or 4 weeks later, and then continue for months, if not for years, number two. And number three, contrary to ECT, a very tight PK/PD correlation. Mm-hmm. You need to have steady state hitting that receptor. If that stops for whatever reason, patient relapse. So the opposite of ECT. And along came ketamine. Ketamine, single dose effect, immediate effect, improvement that are noticeable for everybody. Number two, same thing. 1 dose is not enough. You need to repeat the dose every 3 or 4 days until you get to remission, and then you could stop. And number three, very strong PK/PD dissociation. The half-life of ketamine in humans is about 4 to 5 hours. The clinical effect lasts for 3 or 4 days, exactly like the electroshock. Up until 2010, the field did not understand what's the difference between those black swans, ketamine and electroshock, and then the garden variety antidepressants. Ron Duman and Georgia Gajanian in 2010 identified the difference. The difference is that ketamine induced an immediate growth of synapses and dendritic spines in the prefrontal cortex, immediate, in hours. Traditional antidepressants take weeks to produce that. That was the first biologically plausible difference between ketamine and the others. And that was replicated with ECT. Electroshock does the same thing in rodents, and psychedelics, that does the same thing in rodents. So when you talk about targets, Murali, we can talk about pharmacological binding targets, but in our view, the target is to regrow dendritic spines and synapses in those patients who are depressed.
Yep.
To conclude, one quick one. The question is what about the hallucinogenic experience? How critical that is, okay? Look at the plastogens I'm talking about. Ketamine, ECT, LSD, MDMA. Completely different mechanism of action, suggesting a mechanism of action is not the critical effect. And they don't all produce a hallucinogenic experience. No ketamine, no electroshock, no MDMA. So that's why those of us who are working on this area believe that the biological basis for the effect of ketamine, psychedelics, ECT is the immediate growth of dendritic spines and synapses, and no, for those compounds, the hallucinogenic trip is not necessary.
Yeah, those are very important observations. I think that's really intriguing. But as we think about molecular mechanisms, starting to pin down some of these things, it's still becoming very challenging. I don't know, Jens, you want to comment? There's a lot of omics work. There's a lot of other approaches. But even with all of the technologies, we still are sort of waving our hands a little bit, right?
I think the bigger the disconnect between the intervention and the outcome in terms of what can we interrogate mechanistically in between these two things. The bigger that disconnect is, the harder it is to develop a drug from the perspective of troubleshooting. Right? When your clinical study doesn't give you the results you want, you don't even know where to look.
Right.
And that, I think, has been a really big problem in my 15 years of clinical development timeframe that I have experience with. But to echo what Vikram said earlier on, I think we are getting much more sophisticated translationally in actually being able to, for example, have rigorous EEG data as one example to use for decision making, potentially for patient stratification, as we'll talk about a little bit later.
Yep.
And so the more we can plug in between giving the dose, measuring exposure,
Yep.
and looking at behavioral outcomes, the better off we are, because we all understand that revolution will be iterative, right? And it takes a lot of trial and error, unfortunately.
So Jens, let me put you in the spot. As you know, we have crossed paths while you were at Roche, when you were at Takeda, and representing the Pritzker Consortium, we have for more than 20 years looked for biomarkers for depression, bipolar, and schizophrenia. And as you well know, there haven't been any yet. So, but however, now we're in a period where we are starting to see other type of biomarkers. So like you just mentioned EEGs, and you guys just have phase 1 data that looks quite enticing. What have changed? What is it that didn't provide the outcome on the omics era, which is still evolving, versus these new biomarkers? What is different for psychiatry?
I think there are two different ways to use omics. One is to understand the variability in response to, let's say, a clinical study or an approved drug, for example. Issues there are that the sample sizes tend to be very small, and the actual statistical robustness of the results that you get is limited. And then there is the problem that omics is by definition a high dimensional data set, and you're almost always guaranteed to find something from the perspective of an attractive p-value. But then it's very unclear what you can actually learn from that in terms of the mechanism behind it. You can always derive a statistical approach that you can then try to replicate or even carry forward into a prospectively defined hypothesis or trial. But again, if it's just based on assembling a panel of 20 SNPs or genes or whatever that you don't actually understand what they do, then that is a low likelihood of success. And then the other approach is that you use omics to develop novel insights into disease biology and start from scratch. And the one that most people in schizophrenia will probably be familiar with is for example, the complement, the role of the complement system in not just in the immune system, but also for synaptic pruning. Schizophrenia. That is something that was very strongly corroborated by genetics. But the efforts that actually try to translate that into new mechanisms or new targets, right? That has not yet yielded very much, and that is an extremely long 10 or even 20-year kind of process, which is outside of the area that most of us operate here, right, in this environment.
Laszlo, you want to add something?
Yeah, I just maybe wanted to add another layer to this conversation about, psychedelic-inspired therapeutics. I think there's sort of a couple of ways that you can look at this. I mean, the exciting thing about these agents is that there's a rapid onset of action. So after a single dose or even after a couple of doses, you initiate some kind of a change in the substrate, which is kind of what's been discussed. And so one way to think about this is, how do you improve upon that? How do you make that safer? How do you maybe even develop an agent that doesn't have the hallucinogenic effects, but still get the same level of effect, this very rapid onset? The other piece to it is that maybe for some patients, that kind of a strong effect is going to be required. You're going to need that kind of an effect. But then how do you maintain that effect after you've given a strong psychotropic agent? And I think that's where the idea of at-home use, maybe no subjective effects, very low subjective effects, kinds of agents and mechanisms that do that. So maybe you have to go into the office once or twice for induction, for initiation, but then you get sent home with a pill and you don't have to come back for 6 months or a year. That's a whole really interesting area of research because you've changed the substrate. How do you maintain that change in the substrate?
Got it.
Yeah. Do you have a follow-up on this question of precision psychiatry, because I think it's very important. As we see the direction that many companies taking with endophenotypes or particular symptomatologies, is that the direction that we're going to end up with psychiatry, smaller groups? And if so, what is the implication for commercialization and big pharma? Maybe Carlos, you guys have an example and maybe you can comment.
Yeah, no, indeed. I was really listening with great attention to Eliseo's comment and to Jen's comments. And I think, when we are talking about this kind of precision mechanisms and how we can basically apply precision medicine to CNS, we tend immediately to think in biomarkers. But it was already mentioned this morning that it's a different biomarker, the one that you can stratify a patient, a different biomarker that you can use to warranty target engagement and to find out the right dose. It is interesting because we are developing a drug against LSD1, no LSD, LSD1, which is an histone demethylase. And in this case, precisely what we have seen is that LSD1 inhibition is able to rescue very complex phenotypes on some subsets of the human diseases, like for instance, autistic spectrum syndrome or schizophrenia. And in the case of schizophrenia, to the comments of Eliseo, I think it's interesting to know that there is a very nice independent work done at the Columbia University, where SETD1A is basically one of the genes which have been traditionally involved with high risk of schizophrenia. And when you produce a knockout mice, basically what you get is a mouse which is having a prefrontal cortex neurons with very poor branching and very poor synaptic capability. When you treat these neurons with an LSD1 inhibitor, what you get is basically this kind of sprouting, this kind of branching, and this kind of prosynaptic activity. So, which is basically explaining why in this case, or at least in part, not only that, you recover also the axonal navigation capabilities. So, coming to your point, whatever you need is basically to reprogram neurons to get these connections again, and then you have another mechanism of action like LSD1 inhibition, which could be for some subsets of schizophrenia or some subsets of autistic spectrum disorder, a first way to dissect the efficacy on these particular diseases. Rest to be seen, of course, in the large population, if this is something which is also applying, but coming from your words, if electroconvulsant agents is working on the general population because you are basically producing this kind of enrichment, you could expect that LSD1 inhibition could also work in a general population.
Mm-hmm.
So,
no, that's a good point. It sort of brings into the whole concept. I think this you touched on it, Manuel, about precision psychiatry. And it's a buzzword that's being used quite a bit. But are there clear examples where that's really played out and been successful? I don't know, Vikram, you have a thought on that?
Yeah, well, we've got to be honest. We're not at that level of precision psychiatry that we can offer to a patient an individualized treatment that's very compelling as yet.
Mm-hmm.
But we're moving in that direction, right? We've seen patient segmentation and approvals coming that way, and also some early indications in later stage clinical studies. I mentioned insomnia in MDD example, psychosis in Parkinson's disease, brexpiprazole with agitation for Alzheimer's. It's really nice to start seeing that transition, but we're not yet there. I think if you start looking at things like imaging approaches or fMRI, where you could start thinking about looking at different brain regions and applying more precise approaches, that's going to be really hard to do in the real world. But there's some exciting developments. I don't know if you followed folks like Leanne Williams at Stanford, who's done this gigantic fMRI study in MDD patients. And the clear associations are at different, patients are being segmented by different areas of the brains lighting up, and she calls them biotypes. And, you can see anxiety cluster with depression and other such effects cluster with each other. So we're starting to get to that point. But then the question becomes, how do we translate that? Because ultimately you have to fit within a DSM.
Right.
And so, making that marriage between, patient segmentation and actual ways to diagnose and prescribe drugs appropriately to patients is going to take some time. And as a community, we should really be thinking about how we achieve that.
Yep, that's a good point. Seth, you want to add anything to that, or Jens?
Yeah, the biotype concept you mentioned is interesting because Carol Taminga is one of our scientific co-founders, and we also worked with her to really better understand the patient population, for example, in CIAS that we have to pursue. Maybe a bit of a general comment is that precision is actually an extremely ill-defined term. And I think most people, when they just try to find or look for something that is analogous to oncology, here's your SNP or here's your whatever fluid biomarker test that will tell you if you will respond or not respond, that is largely an unfulfilled promise in psychiatry, and I would argue that it will likely be like that. However, what I found really encouraging is that we are, as a field, more open to pursue either the patient populations that don't respond fast enough, like in the case of psychedelics, but still looking at restoring a euthymic state. But then we're also, for example, in depression, but we're also opening, I think, a whole universe of embracing more the completely unaddressed symptoms of the diseases. We already talked about sleep disturbances, right? With the orexin targeting compounds, you could say something similar is happening for anhedonia. You guys obviously are working in that field as well, as are others targeting kappa. We are targeting cognitive impairment in schizophrenia, and perhaps the best example to your question, Murali, where this actually did play out, although it was a very long effort, is perhaps vortioxetine for cognition in depression, right? That was a really heroic effort. Right. Disclaimer, I used to work for Takeda, but still, I wasn't involved in that program. And I think key for success for future programs will be whenever you can link these subdomains of diseases or these symptom clusters like anhedonia, like cognitive impairment, when you can link them to your actual mechanism and your biomarker that helps you understand that you're on the right track.
Exactly. Yeah. No, I think that kind of opens up the one of the last topics here to cover before we talk about partnerships is around, regulatory paths, right? So, for each of those things, you have a regulatory path, and we don't want to cover all of the topics here, but I know, Carlos, as you're thinking about new indications like borderline personality and other disorders where there is nothing approved, right? You're kind of carving new paths. So that's maybe one example that you may want to touch on. And we don't want to come back again to psychedelics, but again, as you think about the next generation molecules, there is also, paths to be carved out in those areas. So maybe, Carlos, you can maybe have a, 1 minute update, and then we'll move to the next topic.
Well, the regulatory is always a key point. No? At the end of the day, you need to convince the FDA that the drug is providing a significant clinical benefit to the patient and is doing so in a safe way. So in the case of borderline personality disorder, we have a disease which has been very little explored so far, and there is nothing basically approved for this indication, which means that the FDA or the industry in general, even the scientific community, do not have an idea which can be, which might be the best way of measuring a clinical improvement under a pharmacological treatment in a BPD patient. So the community has produced and validated some specific scales. There are also generic scales that have been used for other diseases. And in our case, when we have been presenting the end of the phase 2 data to the FDA, we were having a discussion about what could be the right endpoints for that. And what is clear is that the FDA wants, and this is a very clear position from them, is that they want that the industry measure things that matters on the life of patients. So in our case, for instance, we are discussing on the phase 3 protocol to go basically for one of the points that were on the center of this diagram that you saw before, which is agitation and aggression, which is something that you might find in several diseases, but is one of the most impairing symptoms of these patients. Actually, when you ask the patients, is the first thing that you mention. So FDA was okay on basically on us trying to use this patient-rated, that's another aspect, patient-rated outcome as a primary endpoint. It's true that they were asking us to do some extra homework, but what we can see as a first stance is that there is a constructive attitude, particularly on those diseases where there is no golden standard, where there is no clear way to measure how the patients evolve. So this is a part of the story, our story. But I mean, I think that other biomarkers like ECG biomarkers or basically biomarkers based on facial analysis and so on will come in the future because we'll be able to complement the evidence. But at the end of the day, what we need to be sure that we are saying we want to be a patient-centric industry. So at the end of the day, what really matters is what the patient feels. So if a patient says, I'm feeling better, I'm feeling less agitated, I'm feeling less aggressive, I'm feeling more under control, I can control my stress, I think that this is paramount.
Yep, understood. Any other examples to highlight here as we think about regulator? I don't know, Seth, do you have any comments as you're thinking about getting into the fringes with new mechanisms and approaches?
Yeah, well, I think FDA, at least for us, and it sounds like for other people, has been very responsive if you can prospectively define the problem. So I think extending my earlier comments that a clinical subtype of a condition or a symptom that may be part of the symptoms, exacerbation or like sleep, for example, could be a new target and also the target population. But I think in all of these ways, we're really using clinical science to identify targets as much as biochemical targets. And I think about the history of bipolar. It seems to me that really it was lithium that was the thing that separated what became known as bipolar from other types of depression. So I think that sometimes drug effects can define responsive populations. And, together the whole enterprise seems to be moving forward very rapidly. And I do think that FDA, I mean, they have a wonderful conference, the ASCP, and, every year they are very engaged in trying to advance and speed psychiatric drug development, recognizing the challenges that we face and the shared interest.
Great. So now we're going to talk about the elephant in the room, funding for psychiatry. And, you may wonder, is the glass half full or half empty? And we are in cycles in this space. And if you look at those who receive neuroperspective from Harry Tracy, you will see that at least in 2024, $6.4 billion were invested in CNS. Of those, $2 billion were invested in psychiatry, approximately. But here we have a great representation of public and private companies, and let's hear from them. How do you guys feel is the environment for raising money right now? Start with.
As Manuel, you pointed out in some discussion, the environment is cyclic. It's cyclic. For those of us, I've been in industry for 34 years. I remember when at Wyeth, the CEO was telling me, Eliseo, why are we spending our money in depression? What is that? What about hormones contraception, and that's it. That's the money making. And guess what? In the 80s, SNRIs exploded the psychiatry. So it is cyclic. I am very optimistic. One quick comment regarding the challenges. As Jen said, comparing with oncology, we can't compare with oncology. It's much easier. Tumor cells are all the same. The brain is a much more difficult organ. What you shape in 10 microns, you have a completely different environment. So I think that we are making significant improvements. I think that the market now has been very tough in the last couple of years. I think it's going to change, and our business is a long-term business. We do things that will materialize 3, 4, 5 years from now. So we need to keep that in mind, to have objectives that can endure a bear market, as has been the case so far. But I am optimistic about the future.
Vikram, you are also in the private space. How do you see it?
Yeah, I mean, we did a $170 million round earlier in the year. I mean, we've seen, mega rounds come through as they're known these days, the north of 100 millions. I'll tell you, it's really challenging to go out and raise money out there right now. Even if you're a clinical stage company, investors are extremely diligent, extremely discerning. I mean, the management team has to shine, the products have to shine. More often than not, you're looking at a very defined hypothesis that has to be tested in a very clear manner. Regulatory paths are scrutinized, new mechanisms are really challenging to get funding now. That's not right. We need the ecosystem to actually, support new mechanisms in early stage as well. So I think clinical stage companies with promising targets, we have seen very successful funding of late. I'm really excited for it. I think in our most recent conversations with investors, even despite the more recent setbacks that we've had, and for some public companies that have some large failures, the appetite is there, I can say. Mm-hmm. So the capital is there as well. And like you said, it happens in cycles. I mean, there's no question about the fact that neuropsychiatry is back in the midst of people's minds, especially on the investing side. I mean, if you look back a decade, it was all neurology, mechanistic, approaches for, small neurology indications, large neurology indications. Psych wasn't funded, and now I think the table's flipped a little bit, if you ask me. So I'm continue to be excited for clinical stage funding in psychiatry.
What about public companies like Seth, your company?
Yes, well, Tonix is public, and so we have more of a day-by-day sense of interest in the market. And I can tell you that mid-August, things really picked up. I don't know, not a macro economist, but it seemed like just the specter of lowering rates had a big effect, at least on our liquidity, market price, things like that. So I'm hoping, very optimistic, 2025 is going to be more of the same and more, more interest in the space. But another thing has been the phenomenal success of Karuna. It's great to have stories to talk about like that. And, we feel that, trospium is, half of Karuna's drug is an old drug. And we feel that that's very similar to Tonix's fibromyalgia drug, that, sometimes you can take an old drug and present it in a different way so that the metabolite and parent concentrations are totally changed. The treatment protocol we talked about, short-term, chronic, can be very different. So I think there's a lot of interest in treating these nagging problems. And the business model for chronic diseases is still a very good business model. I think that, there's more of a struggle with, for example, acute pain, where there are only a few tablets per episode, that kind of a thing. But for chronic conditions, I think there's we see a lot of interest from public investors.
Anybody else want to comment on the funding?
I do want to comment that there's a general recognition that the funding cycle has to be large for neuropsych companies. Yeah. So there's deeper investment appetite once an investment decision has been made. So that's a really encouraging sign to see. Investors are typically reserving 2 or 3 checks for their portfolio companies.
Yep, that's good.
Yeah.
Laszlo, you want to
I
add?
mean, I would only add the point that I think in the last couple of years, from the investment side, things have been a bit more conservative. The bar has been raised quite a bit. That's not overall a bad thing, but usually what that results in is, people start to, want to invest in things that are more de-risked, and that tends to be, programs that are in mid-clinical stage, right? Once you have proof of concept and it works, you've taken a lot of the risk off the table, and that's where, when in conservative times, that's where the investments start to shift. But it does kind of feel like it's starting to loosen up a bit in the last six to 9 months. So hopefully we'll see that continue through this year.
Yeah, you mentioned something that is very important, which is, when you have an asset that going to have an inflection point, which is likely the clinical one, it's a little bit easier to get funding. If you are preclinical or you have, number of years before you see that inflection point, I think that the environment right now is a little bit tougher on that regard. Carlos, you were going to say.
Well, no, I mean, I think that every case is different. I mean, what we have seen is, as Eliseo said, a nuclear winter on the financing in biotech in the last 2 or 3 years. So it has been tough, continues to be tough. But I mean, I think that there are signs of really, really promising signs. We have seen a number of funds closing very interesting raisings. We see an interest certainly on the field. And in our case, what we are is basically dealing with the FDA, and we think on our conversations that a number of investment funds, what they are really is waiting for seeing what is going to be the final word from the FDA when we present because at the end of the day, in the public markets, what you are expected to be is basically to have a very advanced asset, a phase 3 asset, which have got the blessing of the regulator, and what are your chances. I mean, if the thing goes well, like Karuna, then you basically have made a success. So investors are cognizant of that. And there is another side effect is that the evolution of the public companies, not only in CNS, in oncology as well, in the last 2 years has been really, really, I mean, they have been having a losing of valuation. So which means that they are very interested in terms of price for the investment fund. So there are a number of positive compounds there that make us to be moderately optimistic.
And the last point we're going to discuss is partnering. And again, if you go to Harry's last report, you will see that about $2 billion in upfront payments in CNS and about 200 and something million in just psychiatry. We know that BMS, Cerebral, Karuna have been very successful. There is unfortunately other companies that have not had those success, and it's not clear yet what the impact on potential partner is going to be. I'm kind of curious to ask the panelists, what is their experience and what is it that the pharma companies are looking for, and Murali can also weigh on this, in terms of what are the milestones that you need to achieve and what kind of partnerships structures you're conceiving or considering right now. Eliseo.
Quickly, taken on the example of Karuna, that seems to be the quantum leap of the last 2 or 3 years. No? We look at xanomeline. Xanomeline abandoned by Lilly, Big Pharma, captured by Steve Paul, that very insightfully took it back to the market, taken over now by Big Pharma again. And I think that this is not random. I think my take is that Big Pharma wants big indications, large indications, and there is room for improvement, but it has to do with the clinical benefit. Lilly thought 20 years ago that xanomeline would not have clinical benefit in schizophrenia. They were wrong. And it took time and effort to do the change the molecule enough to make it the success it is. So the challenge remains for us, non-large pharma, like Laszlo said, the investment in these earlier compounds earlier past that haven't got still yet to clinical demonstration proof of concept.
Yeah. I mean, just to maybe a couple of comments on the pharma side, I think, the exciting part is a number of pharmaceutical companies, big pharmas are coming back, right? I think, they are back in psychiatry. I mean, AbbVie, for example, we are committed in this space and we are not going to be cyclical, right? I mean, we are going to be consistent here in terms of how we are going to be doing partnerships and deals. The business is open and, we are, the business development apparatus is fully functional. Yeah, there are going to be ups and downs, right? Everything we do is not going to be successful. There will be failures. But as an organization, and I think this is true for other pharmas as well, we're going to learn from those experiments. And I think, Vikram, you commented on it. I think as long as there is a clear testable hypothesis and there is a clear way to do those studies, I think if the path is clear, we're willing to take risks, right? I mean, you take the right molecule with the right level of target engagement, asking the right scientific questions, and we do a clean experiment. We want it to be successful, but if it is not, although if you've done a clear experiment, we're still willing to take those sort of risks and do the experiment. So I think that's the way we think about it, right? I mean, for example, the partnerships we have set up early, for example, with Laszlo and his team, again, it's early days, but again, it's a testable hypothesis that, we can take it forward and test it. So, from the pharma side, at least from the AbbVie perspective, we remain committed to this space and we are going to be looking at opportunities where, the science is good, the testable hypothesis is there's a good molecule to go ahead and prove and disprove the hypothesis. And that's kind of the way we look at it. But
yeah, I give you kudos, Murali, because, having people like you in the industry is critical for this to happen. So I think we have time for one question.
I will like to add just one, because that's connecting with the first panel this morning.
Mm-hmm.
When the gentleman who was chairing, he said, well, the investors in a company wants the company to be acquired. This is basically, and it's obvious, which is the most straightforward way to monetize the investment that the investment funds have done. But when you are facing this kind of very complex biology, very complex trials, I think that when you talk about structures, I think that the flexibility to figure out some kind of risk sharing or more creative structures that can solve the questions before you make the big decision, is probably a much more pragmatic approach.
I think we only have time for question for the audience, if there is any. If not, we can continue talking. We have plenty to talk about. Anybody wants to ask? No. So, in the question of partnering, come on, someone else here.
So
yes, you have been on both sides.
I have one comment because I found it very interesting that most of the current generation of clinical stage assets are, as we just discussed, in some way formally abandoned by big pharma. Not all of them, of course, but many, right? Karuna, that's true. Cerevel was the Pfizer asset. We have a molecule from Mitsubishi Tanabe in license because they discontinued their efforts in psychiatry. But that will someday reach a natural expiration date, right? So what's actually going to come after that? If big pharma is perhaps coming back, but as a whole less committed to psychiatry, also for discovery, who's going to make the next or second generation of assets that then maybe we
crazy biotechs.
Exactly. So that's my point, right? So I think many of the companies will have to be more discovery stage also, not just development stage.
Good point, yeah. The investment in discovery has to happen. Exactly. Yeah. I think with that, maybe we should close. I think our time is already run out.
Okay, the timing has been, I have to say, it's very exciting to be in this panel, which every year, if you have been following, thank you, Leo, you have seen changes. And look at these panelists and the companies and what they represent and the biology behind and the stage of the development. It's really, really exciting to see what is happening in psychiatry right now. So thank you, everybody.
Thank you. Thanks to the panelists. Thanks for Sachs Associates to have this.